Welcome back, AP Biology students. A shout out to Joey and Elisa from my first hour AP Bio class who have asked me to make a video on proteins. This is probably going to be long, fair warning. In the future, I'll try to make a couple short videos of proteins. But proteins are super important. They take part in all of our chemical processes in our cells, in all living things, and they're a big structural component of each of those cells. So we want to know a little bit more about them. We should definitely be able to identify functions of proteins in all types of cells, explain how a polypeptide is constructed from the single monomers of the amino acids, compare 4 levels of protein structure, and then analyze factors that affect protein structure and function, because there's a direct correlation between structure and function, and that's kind of an underlying theme all throughout the AP Biology year, is that structure determines function. So let's get going. A couple of things you might not have known about proteins. One is that they make up 50% of the dry weight of living organisms. So take the water away, which is a good portion of it, and the majority of our bodies, or all living organisms' bodies, is proteins. And then there's hundreds of thousands of different types of proteins, all with a different structure, all with a different function, but they have the same basic backbone. So they have a similar basic structure. And that is that they are all polymers of amino acids bound by peptide bonds. So this is showing you the basic structure of the amino acid. You have an amine group, a central carbon atom, a carboxyl group, and an R group. So proteins are involved in almost everything your cells and your bodies do. They are a major factor in your metabolism. So metabolism, your sum total chemical reactions, both building up, anabolism, breaking down, catabolism, is pretty much monitored by enzymes. Enzymes are biological catalysts. Catalysts speed up chemical reactions. Enzymes work by binding substrates at an active site. They might bind 2 substrates and then orientate them close together so they're more likely to react and bond. Or it'll take one substrate and then change its conformation, and when they do that, they weaken the bonds and allow it to be more easily broken. Proteins are also a major component of structure. So we have structural proteins that provide support to cells as well as to tissues. So keratin found in your hairs and nails, collagen found in ligaments, tendons. Transport proteins are used both inside of the cell and outside of the cell. You have transport proteins inside of your cell that move material all the time, as well as carrier and channel proteins that are in the surface, the membrane itself. So carrier proteins may be like a glucose molecule has to move across the membrane. It's going to require a carrier protein to do that. Charged particles, ions, will need a channel protein to allow them to pass through the cell membrane. Hemoglobin is a carrier protein as well. This is moving, it's carrying oxygen within the cell rather than across the cell. And then aquaporins are aquapores, water pores. So in the cell membrane, this is an aquaporin right here. It is a transmembrane that acts as a channel that allows water molecules to pass through. Proteins act in cellular defense. These are antibodies right here. Antibodies are proteins that bind free-floating antigens. So this is a foreign substance that doesn't belong in your body. So the antibodies will bind to that antigen and make it so that antigen can't enter into any of your cells and cause further problems. Proteins are involved with regulation of different body processes, such as the release of glucose or the storage of glucose. So we have signal proteins like the growth hormone over here and the insulin molecule that regulates glucose in your blood sugars. We have receptor proteins on the cell membrane that receive signals. Acetylcholine is a neurotransmitter that tells muscles to contract. And then you have proteins of motion. So this is actin and this is myosin, and they create muscle movement by sliding past one another. So this myosin molecule pulls on this actin molecule, and as it pulls it in one direction, the muscle fiber gets shorter and shorter, thus creating motion. You also have cells, proteins within your cells that move different substances in the internal environment of the cell itself. So this is the basic amino acid structure. Notice you have this central carbon atom, and then you have your amine group in blue, and you have your carboxyl group in green. And then down here, this is a R group. It's a variable group. So if you think of like algebra, you use X to represent a number of different numbers. R is used to represent any of a different set of functional groups. It could be as simple as a methyl group or hydrogens. It could have multiple carbon-hydrogen backbones, but it's going to be carbon, hydrogen, oxygen, nitrogen, and possibly sulfur. So any of those go together in that R group. So each of the R groups have different functional properties, and so they give unique chemical properties to each of your amino acids. So the name amino acid is coming from the amine group and the carboxylic acid group. So that's kind of easy to remember. So here's an example of an R group where you have a hydroxyl. So this hydroxyl is going to be polar, and so that's going to give a hydrophilic property. Over here, we have to find, here's our central carbon, and here's our amine. So this is the side chain here, and this is a whole bunch of methyl. So just carbon, hydrogen, and we know that that is nonpolar. So that is going to be hydrophobic. And then we have some here that have sulfhydryls. So these are going to be able to interact in disulfide bridges, which are going to be really important in maintaining the overall shape of a protein. So this is just a little reiteration, slowing down on that bullet point. All of these amino acids here are considered nonpolar. And if you remember, nonpolar is going to be repelled by water. Water and oil, remember, don't mix. And so nonpolar amino acids will try to orientate themselves away from either the extracellular fluid or the intracellular fluid. The cytoplasm itself is a fluid, right? So the nonpolar amino acids will cause that amino acid to fold inward to the interior portion of the protein. Nonpolar amino acids are all hydrophobic. And you'll notice there's a whole bunch of methyl on these groups. I liked this picture here because it was showing the hydrophobic interaction. So one molecule will fold inward, so will another molecule, so it causes amino acids that are nonpolar to clump together on that interior surface. So here we have some polar molecules. Notice all of these have hydroxyls in them, hydroxyl or amines. And so that's going to make them polar, and we know polar molecules are hydrophilic. Hydrophilic means that they will associate with water. So you're going to find these on the exterior surface of a protein orientated toward the water. So here is a transmembrane protein. This portion out here is oriented to the extracellular fluid. This portion here is oriented to the inside of the cell, the cytoplasm itself. Those are your hydrophilic regions. In here, orientated to the inside of the membrane, trying to avoid the liquids on either side, that's going to be your hydrophobic portion. You also have ionizing properties where the polar molecule may have the tendency to give up hydrogens or to accept hydrogens. So aspartic acid is one of those examples in glutamic acid, and then the ionized form is glutamate. So these were once carboxyl groups, right? So the carboxyl group, remember, has a double bond O-OH. So here you see a hydrogen is missing. So if they're hydrogen donors, right, they act as acids. Here we have amine groups, and remember, amine groups can accept an extra hydrogen. And so if something accepts an extra hydrogen, then they are basic in property. So here you see what might happen is you create ionic bonds between a basic and an acidic amino acid, which causes interaction between different portions of the polypeptide chain, which is one of the things that's going to cause the whole group folding. So we refer to the protein portions as polypeptides. Poly means many, and peptide, it's referring to peptide bonds. So we need to know a little bit about that peptide bond, and you can see down here the peptide bonds. So yes, another bond. We talked about glycosidic linkage with carbohydrates. We talked about ester bonds with lipids. We talked about phosphodiester bonds in DNA and peptide bonds in proteins, which makes it easy to remember. The protein chain is called a polypeptide. So here is your polypeptide chain. It's happening between the carboxyl terminus of one and the amine terminus of the other. So remember dehydration synthesis. One molecule will lose a hydroxyl, the other molecule will lose a hydrogen. The two together will make water, and so we refer to dehydration, taking the water out, synthesis, building something bigger. This is also called a condensation reaction. I want to talk a little bit more about that peptide bond because it's actually really important in the overall structure or shape of the protein molecule. So here you see your carboxyl group, and here you see your amine group. You see the water has left. Now, this carboxyl group, the oxygen, remember, is highly electronegative. So that's going to pull strongly on the electrons of this portion of the molecule, which means you're going to have a negative end and a positive end. Let's look over here. So this carbon is going to be partially positive, and this oxygen is going to be partially negative because it's pulling unevenly. Over here, your amine group, same situation. The nitrogen is less electronegative than oxygen, but it is more electronegative than hydrogen, which means it's going to pull, once again, unevenly on that electron than the hydrogen. So in the end, what you have is a portion of your amino acid that is negative here and here, and a portion of your amine group that is positive here and here. So remember, hydrogen bonding happens between a partially positive hydrogen and a partially negative oxygen or nitrogen. And so this is going to make hydrogen bonding very possible between nearby amino acids. This also gives directionality. The amine group has to bond to the carboxyl group. You can't have amine-amine, you can't have carboxyl-carboxyl. So this gives a directionality to the molecule. It has to orientate itself so that the amines associate with the carboxyl groups. So again, the structure of the protein directly determines the function of the protein. And when we say structure, we're referring to the shape. That 3D structure is dependent on the amino acid sequence. Remember, you have 20 different amino acids. They're kind of like words in our language, and we can put our words together in all kinds of different sentences. So the sequence determines a sentence. That sentence is a gene. So your amino acid sequence is dependent on the gene, and remember, the gene is a portion of your DNA. So this is showing you a few different structures of proteins, clearly different structures, clearly different functions. So collagen that you find in your tendons and ligaments provides strength. Over here we have insulin. Insulin is one of your chemical hormones. Over here we have pepsin. Pepsin is an enzyme that binds proteins. So it has this open space here, and that's your active site. The substrate, this would be some sort of protein. It acts on a peptide bond, pepsin, and so it's going to break those peptide bonds and help in digestion. And then we have the hemoglobin molecule, which has 4 different polypeptide chains all bound together, and they each have what's called a hemigroup. Here the hemigroup contains iron, and iron is what allows hemoglobin to bind to oxygen. So how do we get these overall structures? So first of all, you have what's called a primary structure. So primary structures are simply the amino acid sequence, not in any particular shape. Just think of it linearly. What is the order of the amino acids in this polypeptide chain? So remember, that's determined according to the DNA. So this is going to be the basis of all protein structures. If you affect the primary structure, you're going to affect every structure that is built upon that comes next. Each protein obviously is going to have a unique structure. That unique structure gives it its unique shape. And something kind of interesting is that the sequence will be highly similar within a species. And we can analyze amino acid sequences to determine how closely related two organisms are evolutionary-wise. So here you can see this is the number of amino acid differences in a cytochrome C molecule. Cytochrome C is part of cellular respiration. It's needed to create energy, and all living things have energy needs, and so all living things have cytochrome C. So we can see here the horse and the donkey, very similar organisms, wouldn't you agree? You could almost mistake one for the other. They only have one amino acid difference in the cytochrome C molecule. So that says that they're very closely related. The chicken has 11 differences. So I could very well tell the difference between a horse and a chicken if I looked at it. But the penguin, another type of bird, right? Similar to the chicken. 13 differences from the horse, only 2 differences between the chicken. So we can use the amino acid sequence to determine how closely related two organisms are on the evolutionary tree. The primary structure then associates into a secondary structure. So you'll remember when I was talking about that partially negative portion of the carboxyl group and that partially positive portion of the amine group. So they're going to create hydrogen bonding. And this is called local folding. Local meaning close by. So they'll form either what's called an alpha helix or a beta pleat. And these were discovered in the 1930s, these shapes by Linus Pauling. He's famous for that alpha helix. And alpha is just because they found the alpha helix before they found the beta pleat. So they found the beta pleat next. And this is happening because of interactions between nearby amino acids. So within the alpha helix, it's every 4th amino acid forming a hydrogen bond. And you can see that up here between that carboxyl group and that amine group of a nearby amino acid. The beta pleat, it's when the protein folds back upon itself, and I shouldn't say protein because it's not one yet, it's just a polypeptide chain. So when the polypeptide chain folds back upon itself, and then all along that distance, there's hydrogen bonding occurring. Again, because of that partially negative portion of carboxyl and that partially positive portion of amines. So the hydrogen of the amine is attracted to the negative oxygen on the carboxyl. So all along the length of that fold, there'll be hydrogen bonding. So the primary structure makes the secondary structure possible. It either creates a helix or a beta pleat where it's folded back upon itself. Now, because of that local folding, we have new interactions possible, and that creates our tertiary structure. So we refer to the tertiary structure as whole molecule folding. Secondary structure, local folding, tertiary structure, whole molecule folding. So these happen because of interactions from distant amino acids and typically in globular proteins. So mostly this is happening because of hydrophobic interactions. So the amino acids that are hydrophobic are orientating towards the inside of the cell and inside of the protein and beginning to cluster together. They can also happen because of ionic interactions between positive and negative amino acids, or because of disulfide bridges. Those disulfide bridges are what stabilize, keyword there, stabilize the protein's structure. Because the disulfide bridges are covalent bonds, right? They're a little bit stronger bonds than any of these other interactions. So in this graphic, I liked this graphic because you could see lots of different things happening. Here we have some hydrogen bonding between hydrogens of a methyl group and a hydroxyl of another. Here we have hydrogen bonds between hydroxyls and amines. Here we have some ionic bonding, and then in here we have more van der Waals forces. Down here we have that really strong disulfide bridge. Primary structure of the amino acid sequence starts to fold locally into your secondary structure. Then you have hydrophobic interactions, ionic interactions, disulfide interactions, creating that tertiary structure, that whole group folding or whole molecule folding. And then you have quaternary structure, and that has to occur between more than one polypeptide chain. So this is typically in globular proteins and fibrous proteins like over here. So each of these individual proteins, so these 3 ribbons of collagen, for example, have their own primary, secondary, tertiary structures. These 4 individual polypeptides of hemoglobin also have their own primary, secondary, tertiary structures. So it's not until you get to the quaternary level that these globular proteins actually are able to perform their function. So this is their final shape. And this interaction between polypeptides can be from many of the same things as a tertiary structure. They're due to hydrophobic interactions, hydrogen bonding, ionic bonding, and disulfide bridges. And they can use a little bit of help. So that's where the chaperone proteins come into play. So it used to be thought that chaperone proteins simply aided in proper folding, but more recent research is alluding to the fact that not only do they aid in proper folding of the protein, but they also can help with correcting misshapenness or cutting out misshapen proteins and actually correcting the protein folds. So what happens here is the polypeptide chain would bind with this cap, and the cap then would allow access for the protein molecule to enter, and then that kind of segregates it from the rest of the cytoplasm and puts it in just the right type of environment for interactions to occur between the polypeptides. The closer they are together, the more likely they are to react, right? So let's hold them all together and force reactions. And so that allows for this quaternary structure to occur. Sometimes proteins don't make it to their final structure properly. So remember, a change in the primary sequence, the amino acid sequence, will affect every level that comes after it because it's going to have different R groups with different functionalities. Instead of folding out, it might fold in. Instead of bonding with one portion of the polypeptide, it might bond with another portion or not bond at all. So in some cases, this change in amino acid sequence could be harmful, which is the case in cystic fibrosis. Sometimes they can be beneficial. So if it simply makes an organism of a different color, or let's talk about melanin. If a light-colored organism all of a sudden has a mutation that creates a dark color pigment, and they're in a dark-colored environment, they're going to camouflage with their environment, and that's actually a beneficial mutation. And sometimes that amino acid change doesn't make a big difference in the overall outcome, like whether an organism survives or not. Like an amino acid sequence that changes your hair color or your eye color really doesn't have a factor in the future of your viability. Sickle cell anemia is a good example of when a single amino acid sequence changes the entire protein shape for the detriment of the organism. Sickle cell anemia is also one that is very commonly used in AP biology examples. We'll talk about it during evolution. We'll talk about it during DNA replication, transcription, translation, and we're talking about it now in proteins. So it's a really good one to be familiar with for the entire year. So what happens is you have a single letter change. So one amino acid, which should be glutamine, is changed for a different amino acid, which is valine. Now, glutamine is hydrophilic and should orientate to the outside of the protein. But instead, valine, which is hydrophobic, will orientate to the inside of the hemoglobin molecule. Thus, other portions of the hemoglobin molecule will be on the exterior surface than the correctly shapen hemoglobin molecule. So now different portions are on the outside, which means two whole hemoglobin molecules will be interacting differently. So it causes abnormal interactions between the adjacent hemoglobin molecules, the whole molecules, which is what causes the puckering in the end of the whole blood cell. Another example of misshapenness is Huntington's disease, and this one I get really interested in. I had a cousin who died of Huntington's disease, and so what happens with Huntington's is you have what's called a tandem repeat of CAG. So CAG being cysteine, adenine, guanine, 3 nucleic acids. A tandem repeat means it kind of like stutters. It ends up, the amino acid sequence ends up CAG, 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 CAG. So it keeps adding glutamate in a row where you shouldn't have like a whole chain of glutamate. Glutamate's a polar amino acid, which means they're going to be attracted to other polar amino acids, right? So those glutamines are going to cause lots of different protein molecules of the same kind to bind together. And so that's what you see here. This is what the normal protein should be, but instead it creates what's called an aggregate. So not only is it clumping together other protein molecules, but those ionic interactions are also attracting other ions in the protein's environment. And so then that causes non-functioning neural cells. Prions are kind of similar. So prions are proteins that act like foreign agents or foreign bodies. They act like bacteria or viruses. They're really just a protein, and they're kind of more recent. They were discovered in 1982. They were given the name prion after protein and infection. So they can be transferred from one individual to another, usually from eating infected meat. So it's the same thing as the Huntington's protein, where it's a misshapen, misfolded form. And so it has portions of its protein then that will attract other proteins of the same kind, as well as other elements in its environment. And so once again, it creates an aggregate which destroys that neural tissue. So all of our known prion diseases occur in the brain and in neural tissue, and they are untreatable and they're all fatal. So mad cow disease is the one that we're most familiar with. That's been around maybe the longest amount of time, but it's definitely like the most common in the human world. These affect humans and other animals. The more recent is fatal familial insomnia, so you just cannot sleep. And that's actually a hereditary disease. So those are happening because of the misfolding proteins attracting other proteins. So other than a genetic sequence that has gone wrong, what kind of things can affect the protein shape? So we refer to proteins as being denatured when they no longer have their shape and therefore cannot perform their function. So typically anything that disrupts that tertiary structure, your hydrogen bonds, ionic bonds, or disulfide bridges, will create a change in the shape of the protein. So temperature is a big one. So temperature affects the speed at which atoms are in motion, right? So the higher temperature, atoms, molecules are moving more quickly, and slower temperatures more slowly. So this is going to directly affect the hydrogen bonds, because that hydrogen bond really just being an interaction, the amount of movement between the atoms can disrupt the hydrogen bond and break the bonds. pH is another thing that will affect either hydrogen bondings or ionic bondings. Those ionic bonds that are happening between acidic or basic amino acids, definitely going to be affected by the presence or absence of hydrogens in their environment. And then salinity affects your ionic bonds as well, because they're charged particles that might be attracting positives or negatives from the protein itself. So enzymes are particularly of interest to us, since they're taking part in all of our chemical reactions. Temperature, pH, salinity, pH all have a specific range, a narrow range that they operate best at, and that's because their shape will become disnatured if you're outside of that range. So this is going to affect the secondary and the tertiary structures of the overall protein, therefore affecting the overall shape. So we've talked about lots of different functions of proteins. You should be familiar with all of those examples. We've talked about how a peptide bond is formed. You should recognize the proper orientation and the importance of that positive and negative piece. We've talked about the primary, secondary, tertiary, quaternary structures and how one determines the next. And then denaturation, which directly relates to the structure and therefore the function of the overall protein. So I hope you take this morsel of knowledge, like this little monkey has taken my banana, and store it away for future reference. So that's all I have for you today. Thanks for listening.